that is the process that's occurring, right? You've got DNA and your body uses DNA to make all the things that you are. So this unit, let's turn that down. We're going to start there. So we're going to start with genes.
I'm here, Dr. Godwin.
Gotcha.
Yeah, me too, Dr. Godwin. Maria made it.
All right. All right. All right. I got you both. All right. All right. Okay. Present and accounted for. So we're going to start talking about genes, kind of the nature of them, how they like mechanically, sure, kind of practically function. So I did hit the button. Yes. So we talk about DNA. DNAs are just strings of nucleotides, right? It's just a bunch of ACTs and Gs kind of laying in a big long string, right? So what is a gene? And so there was a lot of research kind of done to this effect. We won't walk through all of that, but basically somewhere in the 20th century, we got to this kind of idea that we've got kind of this hypothesis about one gene coding for one enzyme, right? So we know, right, that enzymes are just proteins and proteins are just chains of amino acids. And so the information that's in the DNA is actually coding for these amino acids that make proteins, that make enzymes, that basically do all of the work making the things that our body needs. Now we know that frequently enzymes are made of multiple polypeptides. And so kind of the more accurate conclusion that we've come to is that every gene codes for a single polypeptide, right? Some amino acid chain. And occasionally it takes multiple amino acid chains to make a full enzyme, right? Some of those enzymes are pretty big and complicated. So we basically have this one gene, one polypeptide hypothesis, right? Polypeptide just being that amino acid chain, right? That forms a protein. Actually, it's more complicated than that, but we're not going to get any more complicated than that for class. We're going to stick at this one gene, one polypeptide kind of concept, right? And so when you're talking about DNA, right, and how DNA becomes a phenotype, right, becomes an organism, we call this molecular biology, right, because we're kind of dealing with the mechanics of life very literally at the molecular level, right? And so the central dogma or the central kind of tenet or theory that underlies basically everything we're going to talk about in this chapter, the central dogma of molecular biology, is basically about the information flow in your cells, right? And so central dogma of biology states that information flows from DNA, right, which is all found in your nucleus, to RNA to protein, right? And then those proteins do stuff, right? So we're going to break apart both of these processes, right? Converting DNA to RNA and then converting RNA or reading RNA to make proteins, right? So we're going to spend time working through these two processes in this unit. So the first process, converting DNA to RNA, is called transcription, right? If you think of like scribes or transcribing writing, right? We're just kind of turning this chain of nucleotides into an RNA chain of nucleotides. So we're transcribing the DNA, right? And then we're going to take that RNA and use it, translate it into a protein, right? We're going to use the information in the RNA to make proteins, right? And so this is the very specific process that is working to convert your genotypes, right? All the genes you have to your phenotype, the actual things that you're made of, right? And so it's all kind of based around this central dogma. Now, for better or worse, we can't really do anything without having at least one small exception. So there is one small exception to the central dogma. Oh, before we do that, this is just a visual example of that. I forgot the slide was in here, right? And so this is the process we're going to be walking through. So whether we're eukaryotic and we've got our DNA in a nucleus, or we're prokaryote with our single little chromosome here, right? We're going to be opening up this DNA, right, to transcribe and make a strand of RNA, which will then be used to make a protein, right? Which is what is happening in all cells and all living things, except retroviruses. Of course, viruses are going to do something bizarre because viruses are weird and not even alive, depending on who you ask, right? So they're kind of doing different stuff because they just kind of exist at this whole different kind of space of interaction, right? And so viruses, retroviruses specifically, which is how they got that name, actually are carrying RNA and convert that to DNA when they kind of infect cells, right? And so we call that reverse transcription, right? Transcription is converting DNA to RNA. These viruses are going around with RNA that they then convert into DNA later. So we call that reverse transcription. So we can account for that, right? We can update the central dogma, right? We've got DNA, right? And as we're kind of growing and multiplying and mitosising and meiosising, right? We're making copies of DNA, that's fine, right? And we can transcribe that DNA into RNA, right? And then translate that RNA into protein. And then we'll just add an extra arrow to account for our retroviruses who are being weird. All right, so transcription, right? Step 1 is transcription. And it is just the DNA directed synthesis of RNA, right? If you have a strand of DNA, you can do a couple of things. You can copy that DNA, right? Because you have a series of nucleotides and you can add their kind of complementary matches, right? A C for every G, an A for every T. You can also do DNA directed synthesis of RNA, which will look fairly similar, right? And so when we do this, right, when we copied DNA, right, in the last unit, we opened up DNA, right, pulled it all the way apart, made a copy all the way down and ended up with 2 copies. Now, when we're transcribing, we're actually just going to be pulling out single genes that we need from the DNA. So we don't got to open the whole thing up. We're just going to kind of pull DNA open a little bit, right? And so I've got my 2 strands of DNA that I've kind of opened, right? Remember, I've got these anti-parallel strands, right? So this strand is going 5' to 3'. This one's going this way, 5' to 3'. And so when we're making RNA, we're going to pick one of these, and it's going to be our template or antisense strand. We'll just do template encoding. So we've got a template strand, and this is the template we're going to use to make RNA, right? Now, I have to kind of think this out. So A, right, if we were copying this into DNA, right, we'd be running down here, A, we'd put a T, right? G, we'd put a C, T, A, C, G, G, C, right? You'd match them up, their little complementary nucleotides all the way down. But we're making RNA, so it's going to look similar, except for T's are going to get replaced with U's, right? So the match for this A is a U, match for this G is a C, T is an A, C is a G, G is a C, G is a C, right? On and on and on and on and on, all the way down, right? And so this is our template strand, right? This is the one we're using to make a complementary strand of RNA. The one we're not using, it's called the coding strand, right? Which is a little counterintuitive. You would think if it's the coding strand, then it's the one you're using. But if we think about kind of the complementarity of strands, right? If this is our template, then the strand that we're making is actually going to look like this one, right? Because both of these have kind of the complementary nucleotides to this template strand. All right, so we've got a template strand that we're using to synthesize RNA, and then the other strand that we're not using is called the coding strand. The other thing that's happening, right, DNA has thymine, T's, RNA has uracil, so all of our T's get traded out for U's, right? So whenever you see U's, you know you're looking at RNA. All right. And RNA can get used for a lot of different things. We're going to spend most of our time talking about messenger RNA or mRNA, right? Because these are the RNAs that are used to synthesize polypeptides, right? That they're used to make proteins, right? But there are kind of a whole mess of different ways we use RNA, right? We're going to basically focus on these 3 types of RNA, but you've also got small nuclear RNAs, small RNAs, signal recognizing RNAs. We might mention them, but we're not really going to spend much time with them. We're going to spend our time up here, right? We've got messenger RNA or mRNA that's used to actually code for protein. We've got ribosomal RNA. You remember back in chapter 3 when we talked about, or chapter 2, whenever cells happened, seems like roughly 5 years ago, right? Ribosomes are made of RNA, right? So we've got ribosomal RNA, and then we've got transfer RNA, which is going to come in and help the ribosome to translate messenger RNA by bringing in the amino acids to make a polypeptide, right? So those 3 RNAs are the ones that we're really worried about. So, got DNA. We're going to transcribe it. But why? Like, how does DNA actually, how does it mean anything? Right, so we're going to talk a bit about the genetic code, kind of characteristics of it, and how the genetic code actually works as instructions, right, for making all of the parts of you. So basic word, right? You can kind of think of DNA as a series of, it's instructions, right? And so then we can think of codons as the words in the DNA, right? So we've got long strings of nucleotides, right? ACTG, GCT, blah, 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 right? Turn it into RNA, trade all the T's out for U's, fine. So a codon, if you're looking at a long string of nucleotides, is a block of 3 nucleotides, right? CCU, ATG, AUG, blah, 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 on down the DNA, right? So we've got a block of 3 DNA or RNA nucleotides, depending on what we're looking at, that corresponds to an amino acid, right? And so that's the trick. Every 3 nucleotides tells your cell, right, stands for a specific amino acid that's going to be incorporated into a protein, right? And that's going to be the translating part when we translate from nucleotides to proteins, right? And so your genetic code is read, right? Literally just read 5' to 3', right? We do everything 5' to 3' in increments of 3 nucleotides, right? NTS is just me abbreviating nucleotides. It's kind of it's a common abbreviation because nucleotides is a randomly long word, right? So you've got little 3-letter words just back to back to back to back to back. And each one of them stands for amino acid that will get incorporated into a protein. Right. And so we're not really worried about deleting bases or anything, but just to kind of see, right? So here's this kind of nucleotide string, right? AUG, CCU, blah, 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 on and on and on, right? So for example, right, AUG stands for methionine, CCU stands for proline, right? And so you can make this direct translation all the way down the code, right? And so that's what your cells are going to actually be doing in this process with your messenger RNA. So if our DNA code, right, is read in these 3 nucleotide increments without any spaces, without any punctuation or any kind of pauses or anything in it, right? Technically, and the genes are just kind of like interspersed throughout the chromosome, right? So you have a gene that's going to be like thousands of nucleotides long. Somewhere in there is a gene. And so you have to make sure you start on the right nucleotide, right? Because if here's where I start, these 3 nucleotides code for some amino acid. But if I start in the wrong place, now I'm reading the wrong code, right? I've got 3 other nucleotides that code for something else and I've messed it up. So the place that you start, right, which is going to be critical, right? I start on this nucleotide and these 3 nucleotides code for some amino acid. That's my reading frame, right? So the first codon in the sequence sets up that reading frame, right? If I start here, then the next 3 on the way, all the way down. Mutations happen though, right? Things get inserted, things get deleted, things get messed up. If a nucleotide, right, if I've got 2 codons here within a gene, and a nucleotide gets inserted, now when my ribosomes read that, they're going to read these 3 and then these 3, including the insertion. So it's going to kind of shift everything, right? It's going to shift my reading frame. I'm going to be kind of looking at the wrong nucleotides. And that's called a frameshift mutation. So insertions or deletions kind of within that genetic code can shift the reading frame one way or another, right? And so it will alter the way that mRNA gets translated, right? Because we're kind of changing where the words are, basically, right? Now, these codons, like I said, specify amino acids, right? Every 3 letters stands for some amino acid that we're going to pop onto this protein. We've got a couple extras, right? They're going to kind of serve as like directions for us, right? So we've got codons that stand for all 20 amino acids, right? That's all of them. There are only 20 of them, as well as stop and start codons, right? So we've got 3 stop codons, UAA, UGA, and UAG, right? And so when your ribosomes or polymerases or whatever see those, right, that's where translation stops, right? My ribosome moves down and is reading 3 codons, 3 codons, 3 codons, gets to this one, it's like, ah, that's the end of a gene. Now I stop translating. Same thing at the beginning of a gene, there's a start codon. And for us and every other living thing on the planet, that start codon is AUG. It's a methionine. You don't really necessarily need to know that it's methionine. But When your ribosome sees a methionine, it's like, ah, this is where I start reading to make a protein. It sets the reading frame, right? I started this AUG, so the next 3 codons, or the next 3 nucleotides, right, are a codon, and so forth. Additionally, kind of an added thing, if you're doing math in your head, if I've got 4 nucleotides and the 4 nucleotides can be in units of 3, and any combination of 3 is going to be an amino acid. Basically, I've got more words than I have amino acids, right? Because it's like, is it 3 times 3 times 3 times 3 words, technically, in our little DNA language. So we say the code is degenerate. Not that it's useless or ill-behaved, but that some amino acids are coded for by multiple codons. But each codon will only stand for 1 amino acid. This actually kind of gives your DNA a bit of playing room, right? Because say arginine is an amino acid. It has 4 codons, different codons that will code for arginine, which basically means that I can occasionally mess up copying my DNA and get a bit of a mutation in there, maybe mess up a letter, and it's still going to give me arginine. It's not going to like completely destroy the protein that I'm making, right? So you end up with something that looks kind of like this. This is the genetic code. Like if you were actually looking at DNA, you could look at DNA and say exactly what protein it's making by looking at this table. You don't need to memorize this table. But you can see here's our methionine, here's our start codon, AUG. Here's our stop codons, these guys. Here's arginine that I just mentioned. And so you'll notice arginine, so if in your DNA, which is then translated to RNA, right? If the codon is either CGU, CGC, CGA, or CGG, all of those codons will result in your ribosome putting an arginine on a protein, right? We call this wobble. We'll kind of come back to wobble. It basically means that you can have mutations that occur in this 3rd position of a codon, and it's not going to ruin the protein that you're making, basically. It's not going to like mess things up too horribly, right? So we call that kind of redundancy or degeneracy in the genetic code. Yeah. So. Additionally, the code is practically universal, which is really, really cool. That thing I mentioned about the start codons, right? So my DNA, the tree's DNA, a worm's DNA, a jellyfish's DNA, all read exactly the same. The language, the words are the same, right? And this is actually kind of one of the strongest chunks of evidence for kind of basically a common ancestor for all life on Earth, because the odds of something as complicated and kind of intricate as the genetic code evolving or kind of occurring more than once, it's really, really slim, right? Now, more practical and kind of applicable to us, right? Because the code is practically universal in every basically nucleus and set of ribosomes on the planet in whatever organism it's in reads everything the same. Because it's universal, genes can be transferred from one organism to the other. I could take genes out of something out there and put it in one of my nuclei and my cell can read it. This is why the COVID vaccine works, because your cells can read the mRNA that those labs created, because everybody's cells understands the same language, right? It gets used in a lot of things. This is one example. This is less useful example, right? So there is a gene in jellyfish that codes for green fluorescence, right? Lots of jellyfish kind of light up and they bioluminesce and they fluoresce and all these sorts of things. So there's a gene that does that, right? It's a protein that fluoresces. You can take the gene out of that jellyfish, insert it into a pig embryo, right? And when that pig grows up, it will express that fluorescing protein because its cells are able to read it and incorporate it into that pig's body, right? Even though it's a gene out of a jellyfish. Yeah. They do this in a lot of stuff. So, and actually this gene, anytime they are studying things, they want to fluoresce to see cells later, they'll just insert this jellyfish gene to make things light up green. So yeah, so that's fun. It gets more practically used, right? Like making stronger crops and all those kinds of more kind of financially beneficial things. We'll talk about those kind of later in the unit. That's kind of one weird example. All right. exceptions, because we can't seem to talk about anything without an exception, but it's kind of like maybe where you would expect mitochondria and chloroplasts because they're weird anyway, right? They've got their own DNA. We absorbed them like millions and millions of years ago. But even then, right, these weird prokaryotic kind of ride-alongs that we've got in our cells, basically the only difference, they have a different stop codon. Otherwise, it's still basically the same language. It's still basically reading the same way. All right, so actual transcription. We've got to get back into polymerases and molecular stuff and all this business. And so as usual, we're going to start with prokaryotes because they're simpler. They've got a little less going on. So they're kind of a nice place to start. So we're going to talk about transcription, right? Converting DNA to RNA in bacteria. And it's going to look similar, but not identical to, right, copying DNA. It's a similar process, right? So we're going to run through these same kind of 3 stages. We're going to initiate transcription. We're going to elongate a piece of RNA, and then we're going to terminate our transcription and release that mRNA to go do its work, right? And then we're going to talk briefly about kind of some things that are a bit unique about prokaryotes because they're going to do some things that our cells actually can't do. Because they're a little simpler, they can be a bit more efficient than we are, to be honest. So guess what? We get another polymerase because we didn't have enough polymerases already. So prokaryotes have 1 polymerase. And we're making RNA. So we were talking about DNA polymerases before to make DNA. It's not even RNA polymerase to make RNAs. That's nice. That makes sense. It's got 2, it kind of comes in 2 forms. So we've got the core polymerase, which is all kind of the brown and green part. You're not going to have to like label it enzyme or anything. But so we've got like the main chunk of the enzyme, which here is kind of all green and brown and stuff. And so that is the portion of the enzyme that can synthesize RNA. Now, the hollow enzyme refers to this core enzyme plus this weird and kind of creepy looking little blue beak pincher situation that's attached over here. All of that together is called the hollow enzyme, right? Hollow just means whole, so it's the whole enzyme, right? And this, along with some characteristics of our RNA, is going to allow us to not need a primer, right? When we were making DNA, you had to kind of put a little chunk of RNA on there, right? RNA on there to get our DNA polymerase started. We have some other things set up in the RNA, and we've got this little kind of clamp guy on our enzyme. So we're not going to have to attach a primer when we make RNA with this RNA polymerase. So what do we need? Conveniently, it's all contained in this mRNA to begin with, or in the DNA to begin with. So in your DNA, you have what's called a promoter region. So it'll be kind of a sequence of nucleotides, and they're kind of like road signs, right? So we're going to talk about being upstream and downstream in DNA. So if here's my 5' to 3', right? We're always moving 5' to 3'. So kind of backwards up that way is upstream, and then the direction we're going is downstream, right? Makes sense. And so say here's my gene, right? I need to make mRNA from this chunk of DNA. Upstream of that is what we call the promoter region, and it's basically going to have the equivalent of little road signs, right? If this RNA polymerase is kind of looking down the DNA, it's going to see this bit of code, right? And it's asymmetrical. It's kind of how your book describes it. But what it basically means is that the code kind of has a first piece that says, oh, you're like 30 nucleotides away from where you need to start. And then it's going to come down a bit, and there's going to be another little chunk of this promoter region. It's like, oh, now you're only 10 nucleotides from where you need to start, right? And so the enzyme's like, oh, okay. And then it's going to clamp on to a start site, right? Because you've got this promoter region. It's kind of tracking downstream. It's like, there's my start site. I'm going to hang on. And then it's going to synthesize mRNA because that's its job until it reaches another piece of the DNA that's going to have a bit of sequence, a series of nucleotides that say, okay, here's where you need to stop. That's the termination site, right? So we've got initiation, elongation, termination, all right here. Right, and this whole region, right, our promoter sequence, start area through the gene to the stop, to the termination site, it's called the transcription unit. Kind of analogous to the replicon in DNA, right? It's the start spot, it's the start the start and the stop, and kind of everything in between, right? So we've got this transcription unit. Right, so it kind of functionally looks something like this, right? So initiation. So here's my RNA polymerase, the hollow enzyme with this pincher stuck onto the front end of it, right? And so the little kind of extra piece is going down the coding strand, right? Here's our template strand. So here's what we're going to make a copy of. And it's going to notice, right? So here is the promoter sequence. It says, oh, you're 35 nucleotides away. All right, we're going to keep scooching on up this template strand. Oh, here's another one. This says I'm 10 nucleotides away. So beep, boop, beep, boop. Here's my start site, right? Gets to the start site, and this theta unit, whatever you want to call it, clamps onto the DNA, right? And opens up the DNA helix. All right. And so instead of having a primer, we've got this thing that kind of takes us along and attaches us at the start site. All right, so once this whole enzyme is stuck on at the start site, we don't need this guy anymore. We're gonna drop him off. Now we get to start transcribing. So that's kind of the initiation process. All right, we're gonna kind of track down, look at our little road signs till we get to a start point. We've got this kind of clamp that gets the enzyme attached on, and then we drop the clamp off and we go on, and elongation will come next, right? So again, as per usual, we're gonna go 5' to 3'. And we're going to add these ribonucleotides, right? DNA is made of diribonucleotides. RNA is made of ribonucleotides. So RNA, right? So I've got my template strand, got my coding strand. We've opened it up just here in the middle, right? And so as this enzyme goes along, right, it's adding complementary nucleotides, and you've got this kind of mRNA that's kind of being produced kind of off to the side as it moves down the DNA. Right? And so as it moves, it's kind of unwinding, going this direction, and then winding up behind itself. So it's only open right where it's making the copy, right? And so this whole situation, right, the coding strand, the template strand, the enzyme, and the mRNA it's making, we call the transcription bubble, right? If you remember the replicon from DNA synthesis, right, which just kind of was like all the pieces that are doing the stuff. Same idea. The transcription bubble here is all of the stuff doing the transcription, right? So our coding, our template, our mRNA, and the enzyme all together, right? And again, as it goes, it's kind of tidying up behind itself, right? So it's unwinding here, it's winding here, kind of moving its way down the DNA till it gets to that termination site. All right. And again, just kind of like the same idea as the promoter and the start sequence, there's going to be a specific kind of little DNA signal, little DNA code for termination, for stopping the transcription, right? And so we're going to go along, we're starting, we're transcribing, like, here's the termination site. The RNA DNA hybrid, right, where they're kind of like making, right, the new RNA code is going to dissociate. mRNA or whatever kind of RNA we've made is going to go off and do its thing. Right, the polymerase is going to let go of the DNA, and the DNA is going to close itself back up. All done. And the way this usually works, like in an actual, or not usually, kind of frequently. Let's go with frequently. One way this frequently kind of occurs, right, and kind of the simplest to talk about is, so in your DNA, the thing that's actually going to stop it, right, so we're transcribing, and we are going to get to this place where we've got loads of C's and G's all in a row. Right, and this is a very kind of chemistry-based kind of physical phenomenon that's going to happen. So we've got all these ATs on our template strand, which means we're going to spit out all of these C's and G's, right? Or I just said that the wrong way around. Series of G's and C's followed by A's and T's, right? So C's and G's are complementary, right? If you had a double helix of DNA, right, the C's and G's are what are binding to each other to make the helix. So if your RNA has this big long chain of C's and G's, they're still going to want to bind to each other, right? Molecules are dumb. They just do whatever it is they do all the time. So this long strand of C and G's are going to kind of come back and like clamp down on each other. And it makes this hairpin, which kind of like hitches up the enzyme. The enzyme's like, well, what the crap just happened, right? It's all like pinched up back here. And because those G's and C's are followed by A's and T's, right? T's being U's and U's being kind of a weaker bond. So C's and G's are really tight bonds. A's and T's and U's are kind of like wussier bonds. So the C's and D's like grab on, make this pinch, the enzyme kind of like grinds to a halt, and then these weaker bonds just let go because they've been all pinched up, and that's what stops the transcription, right? And so then the RNA is released, and the mRNA goes on, finds its ribosome, does its thing. So we can stop transcription that way. Like I said, translation a second ago. All right. Now, weird things that prokaryotes do. So our cells can't do this, right? Prokaryotes can kind of get away. They've only got the one chromosome. It's not as messy. It's not as complicated of a process. And so they actually couple transcription and translation, right? So transcription, making their mRNA, translation, turning that RNA or using that RNA to make a protein. So this is kind of a scanning micrograph of this process, but we're going to look at it in kind of a cartoony sort of form. So here's our DNA. Here's our RNA polymerase kind of trucking on down this DNA, making this mRNA, right? As it goes, mRNA gets longer and longer and longer, right? Now, if you were a eukaryote, this mRNA, right, we would hit our termination site, it would pop off, it would go find a ribosome, do its thing. In prokaryotes, things like bacteria, while RNA polymerase is working its way down the DNA, the ribosomes come on and are glomming onto this mRNA while it's still being transcribed. So it's called a polyribosome, right? So you've got this mRNA with all of these ribosomes stuck to it. And so actually, while the DNA is being transcribed to RNA, the RNA that's already been made is being simultaneously translated into proteins. All these ribosomes are then working down this mRNA to make proteins from the mRNA kind of in the moment, right? All at the same time, right? This is, our cells just can't do this, right? So they have this way of coupling transcription and translation into one process. Additionally, they're just way more efficient than us, just in general. Prokaryotes have operons. So in us, each piece of mRNA codes for a single gene, right? Codes for a single enzyme or polypeptide. In prokaryotes, they're mRNAs, right? The thing, right? That transcription unit between the start and the termination site can actually contain multiple genes, right? And so prokaryote DNA is then organized such that in these transcriptional units, you have all of these genes, and they will frequently be genes that code for all the enzymes in a particular biochemical pathway, right? And so these genes with related functions are all clustered together, right? And so frequently for a single pathway, they can be regulated together then, right? We can kind of do all of this at once. And they really can, right? Because they're already transcribing and translating at the same time. But not only that, so they're transcribing this mRNA, they're translating this mRNA, and they're making all of the proteins for a single pathway, basically in one go, right? Kind of with all the same set of mechanics. And so all of these functionally related genes, they get kind of lumped together in these single transcription units are then called operons, right? This is not how our DNA transcription works, but it's a thing that bacteria can do. Partially because they only have that single chromosome, right? Everything is all together in one place anyway, right? When your genes are spread out across kind of dozens of chromosomes, things are going to get a bit more complicated. All right, so that's prokaryotic transcription. We are going to, we'll see where we get to in eukaryotic transcription. So it's gonna be more complicated because that's just kind of how eukaryotes do it. So we're gonna have different promoters for different polymerases because we're gonna have more than one polymerase. The processing of our transcripts are gonna be a bit different, right? Because we're not gonna be translating them right in that kind of right at the same time. We're gonna have to kind of separate this out in space and time a little bit. And then we'll talk a bit about the difference in the elongation step and transcription step between bacteria and eukaryotes, because because eukaryotes kind of do their own weird stuff, basically. I mean, that's basically all there is to it. All right. 3 polymerases, aptly named 1, 2, and 3. So basically, they're just, the different polymerases make RNAs for different purposes, right? They make different kinds of RNA. So polymerase 1 transcribes rRNA, right? So RNA polymerase 1 basically makes your ribosomes, right? Makes the ribosomal RNA, right? So it transcribes the RNA that will become the ribosome. Polymerase 2 transcribes mRNA and some other stuff, but mRNA, right, being the one that's going to be translated into proteins. Polymerase 3 transcribes tRNA, which we'll be concerned with, and some other stuff, right? So 1 makes RNA for your ribosome, 2 makes mRNA that will actually be translated into protein, and then 3 makes tRNA, which is going to be helping your ribosome make a protein. We'll see that in translation, right? And so each of these polymerases has its own promoter, right? Its own little DNA signal to start wherever it is it needs to start to make the things it needs to start, right? Which makes sense because you don't want your ribosomal polymerase making, I don't know, it would get confusing. So each of them has their own kind of promoter to make sure that they only are transcribing the things that they need to be transcribing. Right? Don't need somebody trying to make your hair color gene into a ribosome or something bizarre like that. So polymerase 2, right, is going to be the one we'll spend a bit of time with because it's making the RNA that's actually going to become proteins. And so initiation then of transcription will be at your polymerase 2 promoter, right? That's where we're making mRNA. That's kind of the point. Now, you don't need to memorize all of these pieces, but unlike prokaryotic transcription, where we just got our enzyme, clamps on, does its thing, we're going to polymerase 2, and then it's got to have a whole bunch of little kind of transcription factors and extra proteins that are going to kind of get everything set up for it, right, at this promoter region. So here's our promoter region. We've got all these kind of helping enzymes that come in, and then polymerase comes in, chomps on at the start site, and then does its thing, right? So we call all of this together the initiation complex. So polymerase 2 and a whole bunch of other transcription factors / enzymes that it needs to kind of help it get started, right? All come together, form this initiation complex that will start transcription of your mRNA. All right, so you initiate transcription, then you make RNA, right? That's like the whole point. So again, nothing seems to be able to work alone in a eukaryotic cell. So we've got an elongation complex set of factors, right? That are all kind of controlled by this CTD, this carboxyl terminal domain. It's just a piece of enzyme, right? All of these are just kind of enzymes that are glomming together to make these big structures that are going to do this stuff for us. So we don't need to worry about the specifics of what this looks like, right? We have this initiation complex that finds our start site, and then we've got elongation factors that come in. Everything's coordinated by this CTD structure, right? And so that CTD is able to recruit elongation and RNA modifying enzymes, because what we're going to see in eukaryotes is that, so we're making this RNA transcript, but we're not going to send that directly to make it into a protein. We're actually going to have to kind of do a bit of like post editing to this mRNA transcript before it gets turned into a protein, right? And so that's this idea that in eukaryotes, the end of transcription is not the end of the mRNA making process, right? Our DNA is just, honestly, it's just messier than prokaryotic DNA. There's all kinds of other stuff in there, right? So you've got this CTD that's kind of handling the elongation and pulling in all these other enzymes to help clean up the mRNA and make it ready to actually be turned into a protein. All right, so what are we doing to this mRNA to make it ready to be turned into a protein? Well, since we're not transcribing it or not translating it kind of like in the moment, right? We're not throwing this mRNA off and then immediately making protein from it while it's still hanging off the DNA, right? we can't just send a little lonely RNA out into the world. It's going to get chewed up by nucleases and all other manner of things, right? So on the 5' end, we're going to put a little cap, right, to make sure it stays intact. And then on the 3' end, we're going to put this little poly A tail, so a bunch of A's, right? Think about it's kind of like a little temporary telomere, kind of like that, right? We want to protect the ends of these RNA pieces. So basically, we're going to put a thing on the 5' end and the 3' end to make sure that the RNA doesn't degrade before we get a chance to make it into a protein. I have 3 minutes. All right, we're almost. I think we're in probably, are we close to a good spot? Yeah, so there is, right? So on the 5' end, we're going to add this little cap, this little phosphorylated cap, and then we're going to add a 3' poly A tail, right? So here's our mRNA in the middle. We've got it kind of capped off and safe, and then we can send it off to get picked up by a ribosome to get translated into a protein. All right, now, perfect. I like ending on a unit. Okay, so when we come in on Monday, we're going to start talking about splicing because capping it wasn't enough, turned out. All right, so that's it. You've got lecture journal for Monday, don't forget.